Chapter 3, Structure and Function of Cells, Screencast 1. This is the first of 3 screencasts for Chapter 1, and this is the last of the chapters that will be covered on the first unit exam, which is scheduled for next week. So next week will be our first unit exam. Also, the review sheet is available. So chapters 1, 2, and 3 will be on the first unit exam. So let's start off with the first concepts, or the concepts associated with this particular chapter. So obviously the focus is going to be on cellular structure and function. So the introduction that we saw previously in chapter 1, that a single cell is the smallest unit of life. Now, we'll also introduce a characteristic of human cells, and that's going to be of the plasma or cell membrane. The second screencast will spend a fair amount of time talking about that. If we look at the nucleus of a cell, that is where the genetic material and DNA is. And we also will consider that to be the control center of the typical animal cell. Now, there's going to be a number of organelles that we're going to introduce. Mitochondria is probably going to be one of the most important. We refer to that as the cellular powerhouse, and that's where the majority of ATP is going to be formed. And remember, ATP, or adenosine triphosphate, that was introduced in the previous chapter, and that is the actual cellular energy currency. So not glucose, not lipid. Our cells will use glucose and lipids to actually generate ATP, and that's covered in the last screencast for chapter 3. Now, an important concept known as the cell doctrine, and this is something I want to make sure that you are comfortable with. We've introduced some of these concepts previously, especially the first. All living things are composed of cells. A single cell is the smallest unit that exhibits all of the characteristics of life, and remember, those were introduced in the first chapter. And all cells come only from pre-existing cells. So cells do not automatically, spontaneously exist or come into life on their own. They all originate from pre-existing cells. Now, there's 2 general types of cells, prokaryotic cells versus eukaryotic cells, and the classification is based on their internal organization. Now, you'll notice that there are some similarities, but probably a future type question would be to compare and contrast the difference between these 2 types of cells. So let's first start off with prokaryotic cells. They have a plasma membrane. Now, an important difference is prokaryotic cells do not have a nucleus. Now, they do have cytoplasm, and that is fluid within the membrane. And also, prokaryotic cells do not have true organelles. A really good example of a prokaryotic cells are bacterial cells. Now, let's compare it to eukaryotic cells. These are more advanced and actually have more advanced structures. They do have the plasma membrane, as do prokaryotic cells, but they do have a nucleus, and that nucleus is actually membrane bound and will act as the control center for that cell. Within the membrane, there's going to be a fluid known as cytoplasm, which is also a characteristic of prokaryotic cells. But a really big difference are going to be organelles. These are membrane-bound structures that have specialized functions. On the first slide, we introduced mitochondria. That would be the powerhouse, and we will see other organelles such as ribosomes, endoplasmic reticulum, and so on. Now, important to keep in mind, all human cells are eukaryotic, as are most animal cells. Now here we can see a figure comparing the animal cell or eukaryotic cell on the left, and the prokaryotic cell is actually on the right. And notice the difference. You can see the fact that in that animal cell, there's lots of substructures that are held by a membrane, a large nucleus that's not always going to be central bound. So don't think that, okay? So there are going to be some cells where the nucleus is not smack in the middle. You'll see that, but let's not get into the trap that that's always the case. On the right, we have that bacterial cell, not nearly as sophisticated, no nucleus, no real membrane-bound organelles. and the genetic material is actually dispersed. It's not contained in any given single area as it is within the nucleus of the eukaryotic or animal cell. Now here we're introducing three types of cells, and what we're going to see on the first one, or A, that is actually cardiac muscle. It's striated like skeletal muscle, but it does have some different properties associated with it. In the middle are nerve cells or neurons, so we'll talk about those a little bit later when we do the nervous system. And the third is actually looking at these cells that are acting as a liner of a kidney tubule. And so there are cells, that's their job. They actually line spaces of tubes and so on. So those will be introduced in the very next chapter. Now, a really important characteristic of cells is their small size. So that's something that I want to make sure that you're comfortable with. And this is important because there's this concept of high surface to volume ratio. And a high surface to volume ratio is a much more efficient way. It's going to allow for better transport mechanisms. It's going to allow the cell to get nutrients more easily and also to dispose wastes. So let's kind of compare what we see here. So I've got this one big old cell here taking up this volume. But look at over here. I've got 8 cells taking up the same space. So I can actually get more done. It's a much more effective and efficient organization overall. This is something that we might see a little bit later, which is an additional way in which some cells will actually increase their surface area with these extensions. So we'll come back to this because it's a very common strategy that we'll see, especially in cells where you see a lot of absorption. So especially in the small intestine and other areas, you're going to see things like that. Now here are basically 3 different images from 3 different microscopes. The first is the light microscope. The second and third are different types of electron microscopes, and they generally have a much greater capacity to magnify particles than does the light microscope. Now, having said that, those of you that are taking Bio 132 or the lab component of this course, those labs begin on this Monday the 14th. Those of you that are in my lab scheduled to meet on Friday, you will be receiving the course information sheet, the syllabus, and also some materials to prepare for our first lab meeting, which will be Friday morning. Okay, so we'll be looking at things related to the microscope. Our work will be limited to the light microscope, but here are some examples or images from 2 different types of electron microscopes. Now here, this is a figure of an animal cell. It's kind of a generic cell. Not all cells will have all of these organelles. This is kind of just like a supercell that's created to demonstrate the organelles that you might find in a animal cell. So that's something you kind of want to keep in mind here. So it just illustrates the organelles within a cell. So this is kind of like a good one-stop shopping image for all common organelles we would expect to find in an animal cell. You will be required to label something similar to this on the first unit exam, which is scheduled for next week. Okay, so not this upcoming week, but the following week. Now, the first significant organelle that we want to introduce is the nucleus. Within the nucleus, that is where the genetic information is stored, and you also want to think of it as the control center of the cell. Now, it has some distinct structural features. It has its own double-layered nuclear membrane, which separates it from the other organelles found within the cell. It has nuclear pores, so that is how something such as messenger RNA, which is produced within the nucleus, will actually leave the nucleus and make its way to the ribosomes. And also, we're going to see the genetic material itself, the chromosomes, chromatin, and so on. And a substructure within the nucleus is the nucleolus. And that's an important area because that's where the transcription of messenger RNA takes place within that suborganelle. Now here just similar images that we saw previously, just larger, so we can actually see in greater detail those structures that are specific to the nucleus itself. Now, the next organelle that we're going to introduce are ribosomes, and quite often I will refer to these as the cellular protein factory. So this is where protein synthesis takes place, and there's going to be two different types of ribosomes that might be found in a cell. Some are free, and those are the ribosomes that are just hanging out, floating in the cytoplasm, and the role of these ribosomes is actually to synthesize proteins for immediate use by the cell. Now, the other type of ribosomes are bound. They are attached to the outer surface of the endoplasmic reticulum. Now, these ribosomes will be producing proteins that will be transported to other organelles or actually exported from the cell to other cells or areas of the body. Now, here we have the endoplasmic reticulum, and this is kind of a general manufacturing center. It is a highly folded membranous network, and there's two types of endoplasmic reticulum. There's the rough endoplasmic reticulum, and that is ER that has ribosomes on the surface. So remember, ribosomes are protein factories, and here we're going to see proteins that will be secreted from the cell produced by the rough ER. Now, we also have smooth ER. This is endoplasmic reticulum that does not have ribosomes on the surface. So one process or thing that takes place within the smooth ER is lipid synthesis. Other things that might be here are some of the lipid-soluble hormones, such as testosterone, estrogen, progesterone, and so on. The other thing that's happening here is the packaging of proteins and lipids, and they're going to be delivered to another structure or organelle known as the Golgi apparatus. Here, I like to think of the Golgi apparatus as a cellular UPS store. I'm not very good at packing things, and so if I want to send something off to family or friends that live far from here, I'll take it to the UPS store, they'll package it, everything, and they'll send it off in its way. So what the Golgi apparatus does, it kind of refines some proteins, okay, but its primary function is a packaging and shipping center, just like the UPS store. So products are packaged into vesicles, and that's the cellular packaging system. And so they will put things into vesicles, and then those vesicles are shipped to other locations within the cell, or actually to the cell membrane, where those contents will actually leave the cell through a process known as exocytosis, which we will introduce in the second screencast. Now, I kind of introduced the concept of vesicles. So these are these membrane-bound storage and shipping containers, and there's four general types. Some are known as secretory vesicles, and those contain products that are destined for export from the cell. There's endocytic vesicles. These contain substances that are imported from the external environment. There are peroxisomes. Peroxisomes actually contain enzymes that detoxify waste products that are produced by the cell. The liver, the largest organ in our body, has lots of these peroxisomes, as detoxification of things is one of the primary jobs of the liver. Another vesicle are lysosomes, and these actually contain digestive enzymes. I quite often refer to them as little suicide stocks, because once again, anything that gets into them is going to be digested by the enzymes that are found within the lysosomes. Here we can actually see how some of this might work. So here this bacteria actually enters the cell. It's basically put within a vesicle. And notice what happens here. Okay, so there's that lysosome that's full of digestive enzymes, and it meets the bacterium. And then the enzymes within the lysosome are digesting that bacterium. And then the residual product, or what's left, are going to leave through exocytosis. Here we can see a peroxisome. So here we kind of imagine what's happening within the liver. Alcohol is lipid soluble, so it easily diffuses into and out of the cell. And then it's going to be broken down within the peroxisome. And then the harmless waste is actually going to be eliminated out of the cell. So it kind of shows you how some of these organelles actually function, peroxisomes and lysosomes primarily. Now, mitochondria is probably one of the most important organelles, and this is not a constant, so not all cells will have the same number of mitochondria. Those cells that are more active than others, such as skeletal muscle, cardiac muscle, and so on, they will actually have lots of mitochondria. And that's actually a training adaptation to exercise, endurance activity, especially where those cells that participate in the exercise will actually develop more mitochondria to produce more energy. So as I said, the number of mitochondria is directly related to the energy requirements of that particular cell. They also have a double membrane, which is a common pattern we're starting to see in some organelles. The inner membrane is highly folded, and we'll actually talk about that a little bit later when we talk about how the cell actually produces ATP, because this is the primary site of ATP synthesis, and that is where cellular respiration takes place. So that mitochondria is going to utilize oxygen to produce carbon dioxide and ATP. So we will come back to that. That's the role, or we'll spend a lot of time on the third screencast talking about that. Now, energy sources that the cell can use are fat and glycogen. So we talked about fat and triglycerides previously, and that's how we store energy for long-term use. So typically the job of triglycerides is lipid storage, energy storage. Glycogen, that's actually carbohydrate storage, and that is short-term energy storage. And if we look at where humans typically have glycogen stores, it's going to be within muscle and liver cells. So that's where we expect to find glycogen. And we kind of talked about that in chapter 2. Now, cells will also kind of have their own structural support system. So they kind of have their own skeleton, and it's referred to as a cytoskeleton. And so what this is just the internal scaffolding which helps that cell maintain its normal shape. Now, there's also some structures that are going to enable movement. So things such as cilia, flagella, and centrioles are some other support and movement structures that we find making up the cell itself. Now, cytoskeleton is made up of microtubules and microfilaments. So microtubules are these tiny hollow protein tubes, and microfilaments are solid fibers of protein. Together, they form that cytoskeleton and the cellular support network. Also, one of the things that the microfilaments are going to play is to help support and anchor organelles within the cell itself. Now, there's 3 types of structures that are specific to movement. One is cilia, and here, okay, we can kind of see that here, and that is not in anemone. This is actually the oviduct, and with the oviduct, it has the cilia, and the role of that is to actually move the egg along through once it's actually been, when ovulation has taken place. But it really does look like a sea anemone, and that's one of the examples I quite often will give what cilia and cells look like. We will also find them in cells lining the airway. So they sweep away irritants and things like that we inhale into our body. Flagella are found only in sperm, and that is the structure of sperm that allows them or provides the swimming ability that sperm have. Now, cilia and flagella have similar internal structures, okay, so they kind of have these overall as far as a tube. And then we have centrioles, and centrioles are primarily involved in cell division, and I'm really not going to spend a lot of time talking about those.